Thank you very much, Mino, for this nice introduction. And again, my apologies for not being able to make it in person, but I came down with COVID and couldn't board a plane. But I'm nevertheless happy to share with you what I would have presented anyway, where I was in Dijon. So I'm going to give you an overview on cross-species genetic method of targets in aging this time. And my talk will be entirely focused on aging. A lot of the data are not published. One important paper will come out at the end of the week in Science magazine. So you can read more about it there. But let me first start by introducing you into the subject. We have been using now since about over 2 decades most genetic reference populations, and the one which we've used a lot is this population called BXD. B stands for a cross between a B6 mother and a DBA father, and there have been F2s generated a long time ago, 40 years ago, and now there's about 185 of these mouse strains available. They're all genetically distinct, but you can, they're actually all clones also, so you can generate many of them. And here you see actually the phenotypic variation in these mice with code colors variation indicated. As such, they are... I have an echo. Okay. There are about 5 million sequence variants, similar to variants in human populations with segregating these mouse. And this means there's about 4,700 genes with protein coding variants, meaning you study about 4,700 mutants at the same time, from which 2,000 have predicted high impact variants. And 6,000 of them, 2,000 of them have predicted high impact variants. And why this population is very particular is because there are 6,000 clinical phenotypes and molecular phenotypes in 35 tissues. And all of them you can look up on these 2 databases. Now, if we organize the phenotypes, the clinical and the molecular phenotypes in these strains, then we can actually create these type of graphs. So we here have the 185 BXD strains, and here every line, there are 6,000 lines in this graph where represent phenotypes, and they are ordered by the labs who made them. And you see a lot of white here. White means that there are no phenotypes collected in this place. And this makes this overview very similar to medical records, electronic health records, where not every patient gets every disease. And so this population is a good way to mimic personal health. But where it stands apart is when it comes to the molecular phenotypes. Here we have 35, and you rarely in humans will have 35 deep biopsies with molecular characterization. For the liver, for instance, you have chow and high-fat diet, you have fasting, feeding, male, female, young, old, 8 different sets of data which complement and which make this molecular phenotype one of the largest phenomes in the world with over 500 million data points. Now, as just one further introduction to genetics, how you can conceive genetics is that we make and pre-calculate matrices where we correlate phenotypes, 6,000, and our genotypes spread along the chromosomes. Every dot here is a significant association. The darker the dot, the more significant it becomes. And you can scan it in a forward genetic way. For instance, say, I'm interested in finding the genes which determine body weight, and you scan along this axis, and then you get what is called a Manhattan plot. And you see here that you have one strong candidate with this ribosomal protein, large subunit 26. We soon will have all our genetic passports. If you don't have it already, many people have, including me. I have a full genome done on myself with 100x. And now I can look, actually, I can say I have a variant in RPL26. And you can scan along this red line and say, what are the phenotypes I have? And I would see, you see body weight again, but no, you also see fat mass and you see VO2 max. So the PWAS, what's called, this phenome-wide association study, is much more powerful because it shows pleiotropy. You don't get one phenotype, but all the phenotypes. Now, given that we have transcripts and proteins in these mice, we can do also transcript or protein-wide association study, or ePWAS. The same principle. You align the phenome, but instead of having here genotypic variant, you have here your transcripts. And again, you can scan them in a forward way, say, what determines VO2 max in the liver? And then you see that VO2 max actually is determined by this transporter, malonate transporter, SLC25A in the liver. And if you know that you have an aberrant expression of your gene in a tissue, you can do the reverse and do what is called ePWAS. And now you see, if you have an aberrant expression of SLC25A10, I have changes in VO2 max, high fat mass, and high body mass. All this you can do yourself when you type in your favorite gene and play a little bit with this website called systemgenetic.org. Now, what I'm going to do in this study is only focus on lifespan, and I'm going to give you life and health span. I'm going to first start with giving you some older data on BXDs, the lifespan in BXDs. Then I give you the overview of the data which we will publish at the end of this week in ITP controls, and then I will talk about an ongoing study in health span and mitochondrial stress in the HTPs. This is old work. And that's also an advantage of the BXDs. You can reuse data which someone has generated before. These are data which are more than 2 decades old when Gerald de Haan and Van Sant actually aged mice, and they saw a twofold difference in lifespan. And you see this indicated here with a pretty good heritability. They were able to detect the significant locus on chromosome 2, but they were unable to identify or pinpoint the candidate gene. Because under this peak, there were 70 known genes. And at that time, the human and the mouse genome were not determined. Work by Riekelt Houtkoper in my lab actually helped to identify mitochondrial ribosomal protein, small subunit 5, as the candidate under this. And how did Riekelt do that? He did 2 things. He said, if my gene is involved in longevity, its expression should change over life. And B, if my gene is involved in longevity or lifespan, it's an essential characteristic, it should translate into other species. And so what he did is he looked at gene expression in the muscle, and if gene expression of MRPS5 decreased by half, he saw that now we get about a 500-day increase in lifespan. Furthermore, if you knock the gene down in worms, he saw a 60% increase in lifespan. And what was interesting, that it was not only MRPS5, it were the entire mitochondrial ribosome, because MRPS5 was the genetic light beacon, the genetic signal, but the entire mitochondrial ribosome behaves similar. And here you see expression levels of some of them, and here you see lifespan of some of this. Now, we published this in 2013 and 2015. There was a paper coming out by a genetics group who also suggested that in humans, MRPS are aging genes, confirming it works in the mouse. Now, let's look a little bit, how does MRPS5 work? 90% of the mitochondrial 1500 proteins or so are made in the nucleus, translated in the cytosol, and then imported, and they will compose subunits of the mitochondrial electron transport chain. The mitochondrial DNA itself encodes 13 mRNAs, which are translated by these mitochondrial ribosomal proteins, which originate actually in these mitochondrial ribosomal proteins originate in the cytosol. Now, if you don't have these mitochondrial ribosomal proteins, what happens is that the mitochondrial proteins fall away, and you have a stoichiometric imbalance in your electron transport chain, which will induce a mitochondrial stress response, abbreviated as MSR. And this response is constituted of the unfolded protein response of the mitochondria, as it induces heat shock proteins and proteases, chaperones. And if it continues, this MSR also involves mitophagy, which you can gauge via pink and parking. Now, we were very lucky because the mitochondrial ribosome is target of a many antibiotics. The ribosome is a target of many antibiotics. The bacterial ribosome is very similar to the mitochondrial ribosome because mitochondria are bacteria which evolved over evolution in our cells. And so we started using some of these antibiotics, and I give you here an example. Doxycycline was the first molecule which we tested. If you treat a worm with doxycycline, you lose the expression of a mitochondrial encoded protein, you get the unfolded protein response, and you extend lifespan. And this is what's happening in C. elegans, but it's also happening in mouse primary hepatocytes, where you see the doxycycline reduces this expression of the mitochondrial ribosome. Heat shock proteins are induced as a response, and actually Xu Wang in the lab tested it in plants, and you see here, if you water plants with doxycycline, they remain more vital and live actually longer. Now, there's one opposite way how you can induce this mitochondrial stress response, and that is by increasing mitochondrial translation. And this we do often with a compound called nicotinamide riboside, where we enhance mitochondrial translation. And here I show you a snippet of a lot of data where I show that this also works. If we use mouse primary hepatocytes, treat them with nicotinamide riboside, you see the induction of the mitochondrial encoded MTCO1 protein. SDHA, which is encoded in the nucleus, doesn't change, but you see induction of chaperones. This, like the induction with doxycycline, has beneficial effects. If you treat a mouse at 700 days of age with nicotinamide riboside, you extend lifespan. You see that you can prevent mouse fatty liver formation. Here you see a mouse which has been given high-fat, high-sucrose diet. If you treat with NR, you remove this. And the same is again happening in the nervous system. If you look at neuronal stem cells, here stained with EDU, you see that actually they stain a lot in young animals. They disappear in aged animals, but if you treat these aged animals with NR, you see them coming back. Now, this brings me to a central paradigm of this talk, is that both mitochondrial disruption, and I showed you inhibition of mitoribosomes, but it happens also in aging or inhibition of OXPHOS, or stimulation of mitochondria, such as with rapamycin or with NR, as I showed you, induce this stress response, this mitonuclear proteotoxic stress. This leads to the unfolded protein response and mitophagy, and this bolsters, strengthens proteostasis, and increases longevity. Now, the question was, can we exploit this therapeutically? Can we give an inhibitor of mitochondria, such as doxycycline, induce this mitochondrial stress response and get an improved mitochondrial homeostasis, or likewise, activate mitochondria with nicotinamide riboside and get the same effect? And so that's what we did. And this is a very recent publication of the beginning of this month, where we actually tested this paradigm in the context of a viral infection. And we tried to use non-antimicrobial tetracyclines for influenza. But let me explain you a little bit about infection. There's three ways how we deal with it. Either we don't deal with it and we die. We fail to clear the pathogen or tolerate the immune-mediated damage. We're mostly familiar about resistance. We eliminate the pathogen. That's what you do when you take an anti-influenza drug. You eliminate the virus. But what's less studied is you can also make the host more tolerant, which reduces tissue damage. Now, the mitochondria are very tightly linked with defense against infection because mitochondrial DNA is so-called foreign DNA in our cells. And so how the cells deals with the mitochondria is very similar as how it deals with infection. And so Adrienne Mottes in my lab set up these sets of experiments where we tested tetracyclines in influenza infection. And we had several tetracyclines. We used doxycycline, but in the meantime, we developed together with a chemist a number of non-antimicrobial tetracyclines, which don't kill bugs, but still retain this capacity of inducing the mitochondrial stress response. If you give a lethal infection to an animal, you see that this animal dies 11 days after influenza infection. If we treat it with either doxycycline or a new antibiotic, half of them remain alive. And you see here, again, that this does not affect resistance because they carry still the virus. There's no change in viral load between the vehicle treated or the tetracycline treated ones. Now, where this compound, which is called 9TB or codename MB14, is interesting is that it does not affect the gut microbiome. It's not antimicrobial. In uninfected animals, here we did a multi-scaling, dimensional scaling of the bacterial community in the gut. You see that there's a perfect overlap of the control and the two tetracyclines. However, once we start treated, we see that the 9TB or MB14 treated ones overlap with the controls, whereas doxycycline clearly is apart. And this is confirmed actually 4 days into the study where you see doxycycline separately, but a new antibiotic doesn't affect the gut microbiome. So that's one way we could do. If we inhibit mitochondrial stress, we can make more robust mitochondria, and we can use this in a disease setting. And we have no data that this works in other diseases, such as Alzheimer's disease, and we're also testing it currently, mitochondrial infarction. But what about the opposite? Can we activate mitochondria, now with nicotinamide riboside, induce these processes, and make mitochondria more robust? And the answer is yes, we can, and it works pretty good. Here we show you some data in a C. elegans model of Alzheimer's disease. These worms overexpress the amyloid beta protein 142 variant. So it's the cleaved variant, the toxic variant, and so there's no effect on cleavage. And if we treat these worms with nicotinamide riboside, we see that we induce the UPR in yellow or mitophagy. We also see that we clear the aggregates in these worms. These worms have aggregates because they overexpress the A-beta protein. We reduce those and they express it in the muscle, so they lose muscle mobility very fast in the red. But if you treat them with NR, you can sustain this. The same we can transfer to a human system, where here we have the SH, SY, 5Y, neuroblastoma cells, which overexpress the Swedish variant of APP. You see here stains with these cells stained with A-beta, you see aggregates, and you reduce them. And Vincenzo Sorrentino, who did this work, also took it into a mouse study, and here he overexpressed he treated APP-PSEN, these are the 2TG Alzheimer mouse, and he stained half brains with thioflavin S, which stains aggregates, which you see here. You see, recognize here the hippocampus. You see that actually there's a lot of aggregates in these mice. If you treat with NR, we reduce them. And we have an effect on functionality, and that's shown here. We did actually a test which is called the fear conditioning test. In this test, you give a mouse, put them in a box, you give it a painful stimuli like an electric shock, which you associate with a cue, in our case, a loud noise. The next day, when you repeat this and give the mouse a loud noise, but without electric stimulus, a normal mouse will freeze, indicated in the red curve, about half of the time. An Alzheimer mouse wouldn't freeze because it doesn't recall that this noise is associated or can be associated with this painful stimulus. But if we now treat the blue mice here, with NR, we bring this back up to normal. Now, another fellow in the lab, Mario Romano, he emitted the hypothesis, perhaps these aggregates are not only typical for neurodegeneration, but are typical for all age-related disease. And he actually looked into muscle tissue, which we had collected before. Actually, Dongryul and Hong Bozan collected muscle tissues from old mice. And here you see we stain them here with an oligomer A11 antibody. In young mice, there's no aggregates. In old mice, there's massive aggregates in the tibialis anterior. If you treat young mice with NR, no effect. But if you treat old mice with NR, no, you see a massive reduction in these aggregates, which is quantified here. This treatment with NR actually also improves mitochondrial function. You see here in the top line that MTCO1 is down in show-treated animals, and that in show-treated aged animals, and you could bring it up with NR. No, this was a mouse study. He took it further and actually also used human primary myoblasts of patients with disease. These are patients with sarcopenia. 3 subjects, very aged, and this is a representative staining. And these are subjects with a disease of the muscle, aggregation disease, which is called inclusion body myositis. It's like the Alzheimer of the muscle. And when you stain these cells, you see that with proteostat, which stains aggregating proteins, you see aggregates in old and in IBM muscle cells. And you basically reduce them by increasing NAD levels, by giving NR, but also by inhibiting PARP, which is another strategy to increase NAD levels. So with that, I would like to conclude this chapter on lifespan studies in the BXDs. And now I would actually like to focus on a new study, unpublished until the, September 30th, on the ITP controls. ITP stands for Intervention Testing Program. This is a study which is sponsored by the National Institute of Aging, where researchers can have their favorite compound tested for anti-aging effects. And the study is actually carried out in a very ingenious way. It's happening in 3 different sites in the JAGS, in the University of Michigan, and in Texas, San Antonio. And it's done in a heterogeneous population, which is a cross between bulb, C57 black 6, C3H, and DBH. And the F2 offsprings are the ones which are tested. And so far, there have been 20 compounds tested in large, very well-powered studies. But what has been never done is look at the survival of the normal ones. And what we did is actually took the tail of the normal mice. These were 2,356 non-treated controls and 920 drug-treated controls where the drug didn't have an impact on lifespan. We have lifespan data in these mouse, we have body weight data, and as I said, we have the DNA collected from the tails, and we've in selected animals we have done liver RNA-seq. But I'm not going to talk about data expression data. Now, the interesting thing, this is a huge study. This is the most powered longevity study in the mouse as such, that we can recapitulate a lot of the findings in humans. If we look, and these are not rocket science, but just we can confirm that females here in the red have lower early deaths than males. These males are not exposed to any of the violence, any traumatic things which we are usually associated with. And we see this very significant difference in survival early on in life in females. Later on, they catch on. The second thing which was very interesting was if we know, carve these mouse up in different lifespan groups. In red mice which die between 6 and 12 months, 12 and 18 months, etc. And if we look, let's focus on the males in the blue at the bottom. We see that the males which die early at between 12 months are the ones which have the highest body weight. The same with these early deaths between 12 and 18 months. And this situation changes gradually if we go along. And you can express this in another way. It's called the hazard ratio. This gives you the risk of dying if you weigh 10 grams more as a mice. It's called the hazard ratio. And you see that if you weigh 10 grams more, you have about a 1.8-fold increased risk of dying in a mouse when you're young. This decreases throughout life. And actually, when you're old, no, you have a negative hazard ratio. That means if you're overweight when you're old, let's say my age, it's a beneficial thing. If you're overweight when you're young, it's a very bad thing. And also you see this in the females, it's significant, but it's much less pronounced, as you can appreciate from this curve. Now you will say, this is mouse, what about humans? So we took this into humans and we used the concept, what's called Mendelian randomization. In Mendelian randomization, you use the relationship between an instrument and an exposure to establish the relationship between the exposure and the outcome. Like, for instance, the instrument is a variant in the LDL receptor. The exposure is your LDL receptor cholesterol. If this is linked, you can say that LDL cholesterol is also linked with coronary artery disease. It's a way to show causality in humans. And we used the 500,000 people in UK Biobank. And what we did is we used here as exposure either BMI or early body weight, and as outcome parental longevity of BMI. Now, if our exposure was body weight at 10 years, these are people which are fat when they're 10 years old, and we looked at their lifespan, we saw that the lifespan decreased if you were fat when you were young. If we look at the relationship between body weight at 10 years and body mass index or body weight in adults, we saw what expected. If you are fat when you're young, you are at risk of becoming fat when you're old. However, what was striking that your old body weight, your BMI when you're old, has no relationship with longevity or a very tiny negative relationship. So also in humans, we could see that body weight, not by itself, but body weight at young age is an important determinant of longevity. And more data are in the paper. The disappointment in this paper was that this was the most longevity study, and we could not find any universal longevity locus if we did scans for males and females in red and blue. You see that we find a female-specific locus on chromosome 3, and for the males in this study, we found no locus which reached the statistical significant threshold. We found a few loci in males which became significant if we truncated the population, actually looked at the very old mice, the mice which survived in if we truncated 80% of the mice. Now, in the last few minutes, I would like to tell you about an ongoing study about health span and mitochondrial stress in the HDP. The HDP stands for Health Span Diversity Panel. And this is even a larger study, which we undertook in our lab. And the question what we ask is, we are all on a different health span trajectory. Like, for instance, the person here in green, he has a high health span. He starts only to encounter disease late in life. Most of us will have an average health span, and some of us will have a low health span. Now, in the European Union, 12% of lives are lived unhealthy. That means the 12%, if you take a person, let's say he becomes 100 years old, he will live from his 90, 88 years unhealthy. If he only lives until 80, he will have the last part of his life living in unhealthy conditions. And our goal is to actually shift this curve to the green curve. So the goal of this study is, can we define health span metrics and trajectories from clinical phenotypes? Can we identify genetic and molecular regulators of health span? And can we improve health span trajectory and decelerate the decline in fitness? So this is not a lifespan study. The previous work was all lifespan. This is health span. And so the study design is the following. We took 90 different strains, 30 BXDs, 30 collaborative founder strains, and 30 inbred strains. We divided them in two pipelines. The first pipeline is called the tissue collection pipeline, the second one where we phenotype them. In this pipeline, we sacrifice mice at 2, 8, and 18 months, either in controlled conditions or after a short-term treatment with NR or DOX. We biobanked samples over 23 tissues, and we have now over 100,000 samples from 80 strains in our freezers. They will allow us to get molecular signatures. In the meantime, in parallel, we pushed mice through a phenotyping pipeline. And this is a quite unique phenotyping pipeline because the mice are housed throughout their entire life in what we call digital ventilated cages, where we monitor home cage activity of the mice and their environment with a 1-minute precision over their entire lifespan. The current recorders is more than 150 years worth of mouse lifespan recordings with a 1-minute interval. Then we phenotyped the mice at 4 months, and at 16 months, they were subjected to an intensive phenotyping protocol covering neurobehavioral, like rotor-rod studies, open field object recognition, echo MRI for fat mass, VO2 max with the Promethean, blood pressure, OGTT, echocardiography, maximal exercise tolerance, and urine analysis. And so, just giving you an example of the DVC, the digital ventilated cages, where we monitor mice 24/7. Here, you see the activity profiles in light and dark phase of 16 of the 19 strains. And we can actually genotype the mice based on their activity profile. You see, for instance, these mice in cluster 3, they are party animals. They actually are already active before the lights are turned down. These animals here in cluster 1, they remain active when the lights are turned off. So they extend, they're like night owls. They stay up all night. And so we have very, very detailed behavioral activity monitoring. But this is just a snapshot. This is cross-sectional at one time. But what we actually can do is we can do this continuously throughout life. And we calculate sample entropy, which we call regularity disruption index, which indicates age-sleep fragmentation. And you see the following here. If you look at this strain, you see here it's behavioral. In young age, you see the purple line, and you see a very big change in behavior when it gets older. And you see this in this PCA analysis, where you see the yellow very different from the purple. On contrast, this strain, the Sky strain, you don't see the separation, and you see that even at old age, at 500 days, this mice has still the same behavior. So this mouse has a fragmented sleep fragmentation, and we can actually say when it occurs throughout life. So this is the power of these data, because we can now apply machine learning and AI algorithms to detect when things go wrong during the lifespan of the mouse. We also have, of course, regular characteristics. Here you see, for instance, body weight evolution in the number of these strains. And here you see the difference plotted between adult and old mice related to phenotypes. And I also would like to point out a few very important differences between mice and humans. Here you see a certain number of mice which become fat. This is the difference. So fat mass increases if it's red. You have other mice which don't become fat, but actually become more lean. The lean mass increases when they age. The interesting thing is that we in the mouse don't see this relationship between fat mass and glucose tolerance. Actually, if anything, mouse become more glucose tolerant when they age. And this is very different from the human situation, where we all know that diabetes is more prevalent in the old and seems to be tracing with body fat mass. And this is very similar to observations published last year by Rafael Di Cabo, who made a very provocative point that OGDT does not worsen upon age in mouse. And we basically can see the same. Now, once we have these data from the tissue collection pipeline and the clinical phenotyping pipeline, we will integrate these data. We can integrate basically the clinical phenotypes and the molecular phenotypes and the genotypes and do GWAS and PWAS. We can use the tissue collection and the clinical phenotypes to do trait correlations, construct networks, and look at model trait correlations to pull out candidate genes, which we can call as validated disease targets or validated disease interception targets, which we then can test in a number of different systems, going from the worm to human genetic population studies. This is an overview of the study. We're currently here. The tissue collection pipeline in young and in adults is finished. The old tissue collection is coming down towards the end of the year, and we have a few animals in the phenotyping pipeline which still continue to be here. And you see that we know of about 150,000 biobank samples from 6,500 mouse. Now, the goal will be that we build cross-species multi-scale models to enable us to build what is called a digital twin of human health. And why do we need the mouse? Why can we not do this in humans? Because in humans, yes, you can do multi-scale biology on the body level. You can do it probably at the organ level in certain biopsies or at the tissue level, but you will never get down deep here. So we use these mouse population data where we have the black box between phenotype and genotypes actually filled up as a predictor to reconstruct digital twins of health, and which will enable personalized healthcare. So this brings me to my conclusion. I show you that GRPs are really a flexible gene and pathway discovery platform, allowing dissection of complex traits and gene by environment interaction, which are not possible in humans, and that these experiments can be seamlessly integrated with human studies, creating a virtuous cycle fueling target discovery. I showed you also that genetic or pharmacological activation of the mitochondrial stress response reduces this proteotoxic stress and increases health span. I also showed you in our ITP study that there is no single magic bullet health or lifespan gene, but it are processes such as mitohormesis, proteostasis, or inflammation, rather than individual genes that determine health and lifespan. And I think these processes can be targeted to interfere with age-related diseases, as I have illustrated here with infection, inflammation, Alzheimer's disease, IBM, and sarcopenia. And this brings me to my conclusion slide with my acknowledgement. Maroon Bousleiman, Gabi El-Alam, and Giacomo did a lot of the work, which is on the latter part of the talk, the unpublished work. I had many old ORX lab graduates, which helped with the rest. And of course, Christina Skonials, my spouse, has been involved in all of it. I thank the collaborators of the intervention treatment program and Sultan Kutlarik for help with genetics. And with this, I would like to thank you all.